Hello, my name is Blake, and I will be discussing the emergence of multi-strain epidemics with an underlying genotype network with collaborators Guillaume Sainonge and Laurent Hubert Dufresne. An assumption commonly found in epidemic models is that one disease is caused by one pathogen, which we know to be largely untrue. Viral pathogens like seasonal influenza, dengue virus, and Ebola virus have contemporaneous strains, meaning that an infection of each could be caused by one of a number of similar yet slightly different versions of that pathogen. These strains do not exist as independent infections, but are interacting. One arises from another through mutation, and the immunity developed against one may protect against another. This warrants the accommodation of multiple strains within epidemic models. The goal of this investigation was to first utilize genotype networks to relate strains genetically within an epidemic model, and then investigate the effects that both cross-protective immunity and mutation have on infection progression. We developed a multi-strain model with an infectious and recovered state for each strain, and infections may progress from one strain to its genetic neighbors as defined by the genotype network, and which nodes represent strains connected by edges that link strains differing by just a single mutation. Cross-protective immune effects are also included, governing the process by which immunity towards one strain protects against others. Here we model cross-protective immunity through a modified transmission rate beta star. Immunity decays with genetic distance between two strains according to a parameter delta that tunes the rate at which cross-protective immune effects drop off with genetic distance, which in this case is the shortest path between two strains in the network. A system of differential equations implemented this model over a well-mixed population. The infection proportions in the endemic state show sequential epidemic phase transitions. Although outbreaks occur for beta above 1, a second transition is observed at the immune invasion threshold, or the point where the infected individuals can effectively infect individuals that have recovered from other strains. Moving over to the right, we see a time series of infections for a lattice genotype network with 25 strains, for all strains and for a handful of individual strain infection counts. We observe chaos-like infection progression when the transmission rate is between 1 and the immune invasion threshold. Both the total infections and strain-level infections oscillate before dampening to the endemic state in time. In the endemic state, we observe localization of infections. Not all strains contribute equally to the infection count. The network on the left shows the infections at each strain relative to the strain with the most infections. In this case, the corners are best able to escape cross-protective effects. The presence of localization in a network can be measured by the effective sample size. Where effective sample size is small, localization is strongest. On the right, we see the effective sample size as a function of transmission rate beta and the level of immunity transcendence delta for the lattice network. We see that a regime exists in blue where immunity transcendence enables localization and cannot be too high nor too low for localization at a given transmission rate. The significance of this model with an underlying genotype network is centered around the rich behaviors it enables once you consider multiple strains. Infections are cyclical and can undergo large-scale fluctuations. The driving factor of these dynamics is the structure of the genotype network, by which the global structure influences infections. Localization is dependent upon multiple factors, including the transmission rate, cross-protective immunity, and the structure of the genotype network. Future work will include the construction of an adaptive coevolution model of strains and ODEs, since genotype networks are not static but continually explore genotype space. A refined understanding of cross-protective immune mechanisms will enable disease-specific modeling, since a strictly decaying function will not be the best model for all diseases. And understanding genotype network structure will depend upon the increased availability of viral genomic data. We would like to thank the Translational Global Infectious Disease Research Center for funding leading to this work.